All right, so we're getting into the next topic here. And our next topic is lipids. Now, lipids have a number of different kind of subcategories in them. We generally think of lipids, we think of fats, but we also can think of steroid compounds as well. So this would be things like cholesterol. These would also be other kinds of natural and artificial synthetic steroids as well. Now, the reason we're going after lipids now, if you can remember all the way back 11 days ago, we were talking about acids and esters and amines. And the reason why we were talking about those is because if we look at the structures of lipids, what we're going to find is that most lipids contain those functional groups that we were talking about. They contain carbonyls. They contain alcohol groups, acid groups, ester groups. So all of those functional groups that we were building right before break, they're all coming back again here in lipids. So just kind of like when we looked at carbohydrates and we said, oh, well, you know, carbohydrates have a whole bunch of alcohol groups on them. And so we talked about alcohols and then we talked about sugars, and they kind of sort of went together. It's the same idea here. Lipids are biomolecules. That means that they are naturally occurring inside of living systems. Again, when we think of them, we usually think of fats. But in reality, they can either be fats, fatty acids, or they can have what is called the steroid nucleus. And the steroid nucleus looks like this. It's a series of 4 carbon rings fused together. Those 4 carbon rings are fused together in a very particular way. It's a 6-membered ring to a 6-membered ring to a 6-membered ring to a 5-membered ring. Because they are so carbon heavy, these are big hydrocarbons, even though they contain some of those polar functional groups, these are nonpolar molecules. The carbon character far exceeds the polar character of these molecules. So they don't dissolve in water. They'll dissolve in organic solvents, the nonpolar solvents, but they won't dissolve in water. The term lipid is derived from the Greek word lipos, which means fat. And where we see these most often, these are what make up our cell membranes, both plant and animal cell. These are heavily featured in what are called fat-soluble vitamins, the vitamins that are nonpolar in nature. And of course, many of our hormones are steroidal hormones, which means they'll have that steroid nucleus as kind of their base. We can break down lipids into 2 big broad categories. We already kind of mentioned these individually. The fatty acids would include things that we think of as waxes, fats, oils, phospholipids, and we'll talk about each one of these kinds of subcategories later on in turn. But what all of these have at their heart is that these substances are largely esters. So again, think about what that ester group looks like. we've got a carbon double bonded to oxygen, single bonded to oxygen, and then we have hydrocarbon groups. Hanging off of either side. But the ester is that middle piece, the carbon that is bonded to two different oxygens. These are substances that when they are hydrolyzed, does anybody remember what a hydrolysis reaction does? What? Not quite. That's hydrogenation. Well, it does have hydrogen. It has something else too. What? Prefix hydro. Water. Okay. So a hydrolysis reaction, we're talking about a reaction with water. Now, to your point, the reaction itself, in terms of the products, looks very, very similar. In a hydrogenation reaction, we have that hydrogen that splits across the double bond. Well, in a hydrolysis reaction, same thing, only instead of H and H, it's H and OH. When these lipids get hydrolyzed, they give us fatty acids and they give us some other molecules as well. Fatty acids, so named because you can see we've got an acid group here at the end. The fat part coming from that long hydrocarbon tail at the end of it. And so the hydrocarbon piece, that's the fatty part. The end cap, that's the acid. It's a fatty acid. Steroids, on the other hand, we see this steroid nucleus that bringing together of these four fused rings. They don't have fatty acids on them at all. And as a result, they don't participate in hydrolysis reactions. So, kind of two key characteristics. The fact that they won't react with water, and structurally speaking, we're looking for that steroid nucleus. Now, in that steroid nucleus, we can have other things hanging off of those rings. And you're going to find, we're going to see carbon groups and carbonyls and a whole bunch of other stuff hanging off of these rings. So if I see that combination of rings together, immediately my mind needs to go steroid. So your book kind of gives this all out as a flow chart. And over the course of this chapter, we'll talk about each one of these groups, at least in part, if not in more detail. But this gives us kind of the differences between each kind of lipid. So what makes a wax different than a triacyl glycerol, which that one's going to be really fun to learn how to say. Well, the main difference is a wax has just a long chain alcohol attached to the fatty acid as opposed to a glycerol group. And we'll talk about what glycerol is in a little bit here. Because ultimately, that's what kind of differentiates them, is what is their structure, what are their attachments. looking at this kind of a glycerol versus this kind of a glycerophospholipid. Well, the primary difference there, one of these fatty acid groups got replaced with a phosphate group. So, as we go through this chapter, we'll look at these differences a little bit more completely, and we'll start to kind of point out the similarities and differences. It's actually a lot less complicated than you think. Okay, quick review. If we're talking about a lipid, which of these are true and which of these are not? Can a lipid contain a carboxylic acid group? Absolutely. That's what we call a fatty acid. So this is totally a characteristic. Can a lipid contain a 4-ring structure? Absolutely. That's what a steroid is. Are lipids soluble in water? No way. Way too many carbons and hydrogens. Are they mostly nonpolar? Absolutely. Remember, polarity is what determines water solubility. So if something is soluble in water, it's polar. If something is not soluble in water, it's nonpolar. All right. Fatty acids. Fatty acids are unbranched carbon chains that have an acid group at the end. So what we are seeing in fatty acids is we're seeing long chains of hydrocarbon with an acid group on it. No branches, no substituents, just hydrocarbon. Now, the fatty acids themselves, that chain length can be super long. We're talking 12 to 18 carbons. Now, depending upon how many carbons are in the chain, that's where we're going to derive some of our different acid names. And so it's going to be kind of a matter of just getting used to using those names. It's not something I'm going to ask you to memorize, but it's something I'm going to ask you to know how to use. So, carbon chain. Because of that carbon chain, these fatty acids are not going to dissolve in water. We already kind of discussed that. We can use the same terms that we used way back at the beginning of organic to describe these hydrocarbon acid chains. If they are all single bonds, so every carbon is attached to another carbon with single bonds, that's a saturated fatty acid, or what we would call saturated fat. If there is at least one double bond, then it becomes unsaturated. Now, we use those same terms way back at the beginning of organic to describe saturated hydrocarbons versus unsaturated hydrocarbons. Alkanes versus alkenes. It's the same kind of thing here. Saturated, all singles. Unsaturated, we have at least one carbon-carbon double bond. Now with fatty acids, triple bonding, not something we see. Not something we tend to see. Double bonding, yes. Multiple double bonding, sure. Triple bonding, not really. When it comes to drawing these fatty acids, because the carbon chain is so long, we generally are going to steer away from this picture here in the middle, the long form condensed structural formula. Because as you could imagine, this is lauric acid. Lauric acid is the baby of the group. It only has 12 carbons in the chain. And it takes up all of the page to draw it. So you can only imagine that if we had 14 carbons or 18 carbons, just how much more fun that would be to draw out every single carbon in the chain. So, well, not quite. There are going to be a couple of homework questions where I'm going to ask you to draw things, just because that's the only way I could get, when we get to reactions, that's the only way I could get to test you on reactions, was draw the products of this reaction. So yes, you're going to have to do some drawing, but your drawings, we've already been working a lot with skeletal formulas. That's where you're going to do the majority of your drawings in skeletal formulas. Yeah.
Okay, so the middle one.
Yeah. They are exactly the same thing. They are all the same thing, in fact. So again, with lauric acid, we're talking about a 12 carbon acid. So here's your acid group. So that's the first carbon. There are 10 CH2s. So those are numbers 2 through 11. And then there's this ending carbon here. That's carbon number 12. So we've got a 12 carbon chain with an acid. Here we've got also 1 plus 10 is 11 plus 1 more. That's a 12 carbon chain with an acid. This 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 carbons with an acid. And this one we'll number like we would if we were actually trying to name it. And sure enough, we get the same exact result, 12 carbons with an acid group at the end. Now, you can see, I've kind of warned you about this since the beginning. There's a reason why we tend to favor the skeletal formulas. Once we can kind of understand what it is and what it means, it takes up way less space to draw that than it does to draw this. And it's not the same as those as well, but we can kind of see everything. Now, as far as like the homework goes, in the homework, there is a chain tool here. So if you click and drag, you can make it as big or as small as you want. And you can see following your cursor, it'll tell you how many are in the chain itself. So if I wanted to draw that same 12 carbon chain, there's my 12 carbons. And then I can just change the bonding here on the end. And there's my acid. So it's not quite the torturous hell that you're envisioning, where you have to click each one of those carbons in. If you use the chain tool, it'll allow you to draw, and it can start from anywhere. So if I started to draw something else and I wanted to draw a chain off of that, I could. So when we're getting into triglycerides, other compounds where it's many different branches, you can do that. Sean, you got a question? Okay, well, the reason the O is turning red here, how many bonds do we usually have around oxygen? Remember, oxygen's this. So it's already got 4 in pairs. It can only make 2 bonds, but our structure's showing 3, and the programming's going, hold on, wait a second, that's not right. So it's trying to clue you in to you've made a mistake in the structure. And so if you change that back to a single bond, the red dot goes away because now you've fixed the error. What? In that scenario, no, this would be an ether, and this would be an alcohol. Remember, for an ester, you have to have a carbonyl group. You've got to have the carbon double bonded to oxygen and the additional oxygen. So if that became a double bond, now it's an ester. All right, so there are a lot of different ways that these get represented, and you'll see all of them in different ways as you go through the book, as you go through the homework problems, and so on. Here's kind of the important stuff as far as fatty acids go. Fatty acids, basically, there are 4 primary types. There's lauric acid, there's myristic acid, there's palmitic acid, and there is stearic acid. Now, again, don't get too bogged down in the names and whatnot. This is an example of something I would give you on a quiz or an exam. But what you do need to understand is what it is saying. So the notation here, what does 12-0 or 14-0 or 18-0 mean? Well, the first number, that's the number of the carbons in the chain. The second number, that's the number of unsaturations, the number of double bonds. And so in this case, all of these are .0 because they're all saturated fats. They're all saturated fatty acids. Lauric acid, the 12 carbon acid we talked about in the previous slide. Where do we find it? It's actually found mostly in coconut oil. Myristic acid, we don't see as much but it is one of the oils that is present in nutmeg. Palmitic acid, it's 16 carbons. We find it primarily in palm. So it's one of the things in palm oil. There are several. And then stearic acid. Stearic acid is found in animal fats, 18 carbons, completely saturated. You can see here melting points increasing as the chain length increases, and that has primarily to do with just when you're dealing with nonpolar molecules, there is usually a relationship between the mass of the structure and how well they interact with each other. The bigger it is, the more they tend to attract each other. That brings up that melting point just a little bit. And so as we add more carbons, we're adding more mass. We're getting slightly bigger melting points as a result. All right. Common properties of saturated fats. Because there are no double bonds, and all of those bonds are all single bonds, and they're all kind of flexible and moving, they can fit pretty close together. They can pack pretty well together. From an intermolecular forces standpoint, remember we talked about dispersion forces? Because of that packing, we get decent dispersion forces. And those better than average dispersion forces raise our melting points, and these substances tend to be more in the solid phase at room temperature as a result. Again, if you go back to these melting points, you can see coconut, coconut fat, boric acid, 44 degrees Celsius. Yeah, that's higher than room temperature. Melts pretty at relatively low temperature. So if you buy coconut, coconut oil usually has some solid and liquid kind of together at the same time, and this is why. Naturally, a solid, mix it with some other stuff, it kind of melts a little bit easier, has a relatively low melting point anyway. Animal fats, melting point 69 Celsius. We see these in the solid phase. Really only see them in the liquid phase when we're cooking. So we can see, because of the way that these structures are, we think about the way that those bonds are, that's where that packing's coming from. These chains can get really close to each other because of that packing, because there is no kink in the line. Remember, when we make double bonds, start having to look at that cis versus trans. Well, in a cis orientation, we've locked the chain in one direction. In the trans, we've locked it into a different orientation. It's going to get harder to pack them in closer to each other because there's no flexibility. There's no movability inside of that molecule. That double bond is fixing the chain to either go in one direction or a different direction outside of that bond. And it doesn't seem like much, but it's enough to notice. So when we introduce a double bond into a fatty acid, we call it an unsaturated fatty acid or an unsaturated fat. If there is only one double bond, we call that a mono unsaturated fat. If there are more than one, we call it a poly unsaturated fat. And so if you're hearing these and you're saying, that sounds familiar, it sounds familiar, that's your food label. Look at the nutrition facts on any food label that has fat, and it's going to break down the fat into 3 categories. Saturated, monounsaturated, polyunsaturated. Especially if the product itself has a good bit of fat in it. Things like butter, margarine, oil spreads, those kinds of things. They're gonna say that because there are different health effects associated with each one. Now, as far as the naming here is concerned, remember that the 16 carbon 0 double bond chain was called palmitic acid. Notice the name of this one with 1 double bond. Pretty close to the same, you just change that ending slightly. So instead of palmitic acid, it's palmitoleic acid. That oleic acid piece, which is what stearic acid turns into, oleic acid, is the indication of that double bond. Look at what that melting point is, though. Both of these are actually melting lower than room temperature. Room temperature is somewhere between 20 and 25 degrees, depending upon how warm you keep the room. Palmitoleic acid melts at 0. It's the same as the freezing point of water. Oleic acid melts at 14. Also well below room temperature. So that one little double bond, now compare. Palmitic acid melted at 63. Palmitoleic, 0. Stearic, 69. Oleic, 14. That one little double bond had a massive impact on the melting point. And everything that we were talking about with packing and stacking here, because of that kink in the chain, we can't stack nearly as well. Now we have to stack around this rigid piece. And it becomes a lot harder to do that. For polyunsaturated fats, again, we're seeing multiple double bonds, at least 2. The naming here, don't get too caught up in what the naming actually is, but understand that as we went from 1 double bond to 2, we're talking oleic acid, melting point of 14, linoleic acid, melting point -5 with 2 double bonds, linolenic acid, -11, it's got 3 double bonds in it. Each one of those double bonds adds more kinks to the rope that make it harder for us to stack them together. And arachidonic acid, this is our first 20 carbon example, has 4 double bonds. Its melting point, -50 degrees. We're not going to see it in the solid phase. With some of these, yeah, some of these we can freeze. -5, -11. Most common freezers can get that low. If I put these into my freezer, I don't know why I would, but if I did, I would see that they would solidify. These oils would kind of take on more the appearance, consistency of like a soft butter or some kind of a spread. But they melt really quickly too. Because we're dealing with double bonding again, we have to bring out our cis and trans terms again. So cis and trans, they mean the exact same thing that they meant before. A cis isomer, again, meaning the same side, the chain came in from the bottom and is exiting toward the bottom. So that's what makes that a cis. In the trans isomer here, it came in from the top and exited on the bottom. Opposite sides of that double bond, that's what made it trans. So the words still mean the same thing. Just like saturated and unsaturated mean the same things here as it did before. Now, based on these structures, which isomer would we expect to have a bigger impact on melting point? Which one's going to have a harder time stacking together? The cis isomer or the trans isomer? All right, why?
Because it's still in the Victorian style. It goes up and then down.
So you're saying the cis would pack easier? Okay. This is more a curiosity. I want to see where everybody's head's at right now. So which one would be easier to pack? Which one would have the higher melting point? Okay. How many say cis would be easier to pack? How many say it would be trans? All right. You're right. They both have a kink in the hose, but this one, because it is trans, that kink is following the natural progression of the chain. So it's going to look a lot more like a saturated than an unsaturated fat. Now it's still, that kink does reduce flexibility. which is going to make packing a little bit harder because you're not going to have the ability to, those bonds aren't going to be as flexible to move around. But it's certainly going to be better than this. This is like trying to stack foam noodles. You put one or two on top of each other, eventually the whole thing's going to fall over. So yes, we would anticipate that the trans oleic acid would have a higher melting point than the cis would. And that's exactly what the next slide was all about anyway. That cis isomer is going to have more trouble because of that extra bending that it does. As a result, these cis conformations tend to be more liquidous at room temperature. We have less of a chance of finding them in the solid phase. All right, let's talk about essential fatty acids. So again, we're starting to personalize this a little bit. As human beings, we are capable of synthesizing some of our own fatty acids. Our bodies are able to make them based upon just kind of the normal habits of metabolism and cellular function. And we primarily do it by reprocessing carbohydrates, reprocessing other fatty acids to create different ones. What we can't do, however, is we can't make certain kinds of fatty acids, in particular, polyunsaturated fatty acids. So linoleic acid, linolenic acid, arachidonic acid. We can't make them. Can't make them on our own. And like many other things, if we can't produce it on our own, that means that it has to be part of our diet in order to get it. You've heard of essential minerals, essential amino acids. Well, essential fatty acids exist as well. And they're all essential for the same reason. Our body can't produce it, so we have to intake it another way. Whether it's through our diet, through supplement, through vitamin, whatever it is. So the essential fatty acids are those that we cannot generate ourselves. And in particular, these polyunsaturated fatty acids are the ones that we find ourselves the most concerned about, most concerned with. All right, let's summarize and check. This is the structure for oleic acid. Let's go through each of these questions. Why is this molecule an acid? Matthew? Yeah, it's got an acid group on it. That's why it's an acid. It's got an acid group on it. How many carbons are in oleic acid? $18. Okay. Now, this is one of those usage things. As you get used to using it more, you're not going to have to count it as much. So stearic acid is 18 as well. Oleic acid is the one double bond derivative of that. It's got 18 carbons also. Is it saturated, monounsaturated, or polyunsaturated? Yeah, it's mono. We've got a double bond, which means it's unsaturated. The fact that we only have one of them means it's monounsaturated. Would we expect this to be solid or liquid? If it's unsaturated, we should anticipate it being liquid. Even if it is trans oleic acid, again, that double bond is enough to just make it still more difficult to stack. So even though it's got 18 carbons, that double bond is going to significantly reduce the intermolecular forces and lower the melting point. Would we expect it to be soluble in water? No. We wouldn't expect that for any lipid. All right. Before we leave this particular part of the chapter, we want to focus on just a couple of common products, body product things that come out of this variety. So icosanoids. These are cyclical compounds that come from the convergence of arachidonic acid. So you can see what happens here is this you create a ring structure on the end. Now, different compounds can come off of this, but the reason why we are, the most interested in these is that first category, the prostaglandins. These are hormones or hormone-like substances that exist in your body, and what happens? These in particular are related to pain centers in your body. So you whack your thumb with a hammer, it hurts. One of the reasons it hurts is inside of your body, as a response to the stress, arachidonic acid inside of your body starts to turn into these prostaglandins. And depending upon, whatever circumstances there are, we can get these different substances, PGE2, PGF2 alpha, whatever. So prostaglandins are hormone-like substances that are produced in our cells. We also can see them referred to as eicosanoids, and they are derived again from arachidonic acid. Arachidonic acid, that 20 carbon polyunsaturated fat. How they differ from each other is primarily not here, but here. That 5-membered ring at the far end, that's where they can change. So if you look, the chain part, the non-ring part, is the same for both of those molecules. Where the difference is this one's got a ketone group on it, this one's got 2 alcohol groups on it. These hormone-like substances have a lot of different functions inside of our body. Talked about it already as kind of like a pain receptor, but we can also see there are relations to blood pressure. There is relation to contraction and relaxation inside of the uterus. There are other kinds of functions as well. Like I said before, in that scenario where you whack your thumb with a hammer and you form these prostaglandins in the process, it hurts. And the reason it hurts is that these are what cause inflammation and pain in the area. It's the presence of these molecules now in your cells that cause that inflammation. To combat that, non-steroidal anti-inflammatory drugs, better known as NSAIDs, work by basically shutting down this reaction. So they introduce themselves into the cells and they prevent the arachidonic acid from converting to these other kinds of hormones. And that's ultimately what stops, or at least lessens, the pain. Right. One other thing that we want to make mention of, a lot gets made out of fatty acids, in particular, what are called the omega family of fatty acids. The kinds of fatty acids that we find in unsaturated fats, in particular in vegetable oils and certain kinds of fish, are generally accepted as being more beneficial to your health than things like saturated fats. So when you get into that big debate kind of with a doctor or a dietitian about what kinds of foods you're eating, this is usually where that conversation ends up leading is, well, what kind of stuff do you eat? What's your diet like? What do you cook with? And these are why. Now, there are different kinds of omega fatty acids. You've probably heard a lot said about omega-3 fatty acids. These are kinds that are found in a lot of fish. In vegetables, we see a lot more of what are called omega-6 fatty acids. Now, omega-3, omega-6, this sounds like a lot of, well, how do I know which one is which? It's actually easy. Anybody know the Greek alphabet? Right?
No.
Okay, you don't need to know the whole Greek alphabet. Generally, you only need to know a few letters of it to be any good at science. You need to know the first letter, alpha, beta and gamma, and the last letter in the alphabet, omega. Because it's the last letter in the alphabet, it's also the last carbon in the chain. So remember how we number acids? We always number at the acid group first, and then work our way to the other side. Well, when we're determining omega blank fatty acids, we go the opposite direction. We start at the end of the chain, and we count the number of carbons till we get to the double bond. So for omega-6 fatty acids, that double bond is on the 6th carbon from the end. That's what makes it omega-6. If it was omega-3, it'd be the 3rd carbon from the end. If it's omega-12, it'd be the 12th carbon from the end. Alexis? There's no need to. So with fatty acids, these are all common names. So when we say linoleic acid, it is understood, okay, this is going to be a 16 carbon, 18 carbon chain, and it's going to have double bonds here and here, because that's where the double bonds in linoleic acid are. And it's omega-6 because if I count from the end, that first double bond is on the 6th carbon. So unlike a lot of the naming that we've done up until this point, where it's very systematic, it's this, then this, then this, figure out what functional group there is. With fatty acids, common names tend to dominate because there isn't as much variation in what could be there. It's not like we're going to have a 3-methyl hanging off of this fatty acid somewhere. The hydrocarbons are completely unbranched. The only variations in them are the positions of those double bonds. And again, if the position changes, we have a new name for that particular that fatty acid anyway. No, it'd be something else. So again, omega-3 fatty acids, same idea. Got a long chain, double bond on that 3rd carbon from the end. Now, why do we care about omega-3s? Well, they are shown to reduce blood clotting, or at least reduce the possibility of blood clotting, because they can act as kind of a barrier between blood platelets to keep them from sticking together. And so where this becomes more important, especially, is in heart patients where you might have cholesterol issues that are causing the arteries to become smaller, closing up because they're blocked with cholesterol. In patients with hypertension, high blood pressure, for similar kinds of reasons, usually. The possibility of blood clotting and platelets sticking together does increase. And so in these kinds of patients, upping fatty acid consumption in terms of omega-3s, using supplements like fish oil or krill oil, can help with that, because They can keep those platelets a little bit further apart and less likely to stick together. All right, we're going to dip our toe in the water here with waxes and triacylglycerols. And I'm going to tell you already, I stumble over that word. I stumble over some of these words. Triacylglycerols have a more common name. You've heard of triglycerides. They are the same thing. A triacylglycerol, that's kind of the more formal name for what it is. So if you're looking at like a reference page or a textbook, they're probably going to refer to them as triacylglycerols. If you look in like a health aspect, if you get a blood panel taken and they do a lipid panel on you, figure out how much blood cholesterol you have, you're not going to see triacylglycerol on there. You're going to see triglycerides. And that's what these are. So we're starting to get into the kind of subsetting of these fatty acids. And so we're looking at what makes a fatty acid subcategory unique. So waxes. Waxes are esters. You can see it right here. There's my ester group right in the middle. On one side of the ester group, I'm going to have a fatty acid. On the other side of the group, I'm going to have an alcohol. And in both cases, those chains are going to be long. So we're not talking about ethanol here and maybe a 5-carbon acid there. We're talking 14 to 30 carbons on either side. Now, primary function of these waxes, these are natural waxes. This is the stuff that you find on leaves to act as a water barrier. Keep water from exiting the plant cell. Also prevent too much water from getting into the cells as well. But again, we're looking at an ester here, fatty acid on one side, alcohol on the other. The other primary group that we were going to look at in this section are the triglycerides. Again, triacylglycerols as well, same thing. Triglycerides are the product of a glycerol molecule reacting with fatty acids. So glycerol looks like this. I've got a 3-carbon chain with 3 alcohol groups on it. Three-carbon chain with 3 alcohol groups on it, 1 on each carbon. Now, what happens in each of these cases, they're going to undergo an esterification reaction. We talked about esterification reactions last chapter. The idea that we've got a hydrogen and a hydroxyl group coming together to form this ester and water. Well, the only difference between an esterification we covered last chapter And this kind of esterification is that it happens 3 times. And so what I end up getting is a 3 fatty chain molecule. 3 ester groups, all on the same molecule, all on this one kind of compact. That's where triglyceride comes from. Tri from the 3 fatty acid groups, glyceride coming from that glycerol backbone that's holding them all together. And that's where we're going to stop for today. So for tomorrow night, there is some work to be done as far as a post lecture is concerned. There are going to be some minor questions that are going to ask you to interpret graphs like these. But for the most part, you know, if you do run into stuff that we didn't explicitly talk about here, you should be able to kind of read between the lines based on what we've talked about in the other things. Bring those questions with you on Thursday if you really do get stuck, or send me an email or a text. I'll be glad to help you. Have a good day.